Hello, my name is Delong Meng, and I'm happy to share my project to reproduce this RefDNN paper. Drug response prediction is a binary classification problem. It is important because the goal of precision medicine is to develop personalized drug treatment plan for different patients according to their molecular background. A simplified version of this problem is given a certain drug represented by its molecular structure and a certain cell line represented by its molecular features, we want to predict whether this cell line is sensitive or resistant to this drug. Two different data sets are used in this study, CCLE and GDSC. Here I show an example of the format of the data set. For each sample, you're given one specific drug, one specific cell line, and the response. Basically, one sample is one drug cell line pair. CCLE has over 5,000 of these kind of pairs, and GDSC has around 200,000 pairs. They also have different numbers of unique drugs and cell lines. The study proposed a RefDNN model. Again, you have the cell line, you have the drug as the initial input. The key of this RefDNN model is that they define a set of so-called reference drugs, which by default could be just all of the available drugs. A cell line represented by the gene expression profile is first converted to this n-dimensional vector using an elastic layer, with each load representing the probability of this cell line is resistant to each drug of the reference drugs. Note that these values will be used to calculate partial loss of this elastic lag part by comparing with the ground truth. There are several hyperparameters to tune for this component. On the other hand, a drug represented by a vector of fingerprint is converted to n-dimensional representation by calculating the similarity of this drug to each one of, again, the reference drugs. Then the cell line and the drug representations will be multiplied to get this input layer for a simple fully connected neural network with two hidden layers, and the output is the final prediction of the drug resistance. Note that this output will be used to calculate a second part of the loss. So there are two parts of losses and two optimizers here. Together, this total loss is used to calculate the gradient and update the parameters. In my ablation class study, I actually removed this partial optimizer and the partial loss to investigate its contribution to the overall performance. In this DNN part, there are also some hyperparameters to tune and some others are predefined. So in this study, Bayesian optimization and nested cross-validation is used to select the hyperparameters and evaluate the prediction performance of different models. So specifically, you have 5 outer loops. Within each loop, you search through 20 different combinations of hyperparameters. And within each hyperparameter combination, there are 3 inner loops. At the end, for each outer loop, the best set of parameters will be used to train the model and generate the final performance metrics. Here are my results. Overall, I was able to reproduce most of the original results of this paper and confirm their main conclusions. Here I show that the RefDNN model outperforms other Cas9 models as well as my test model. I further reproduced the NIV1 group cross validation and biomarker identification of this paper. For details, please refer to my report.